All right, everyone, let's get started. So it is my pleasure to welcome Stas Shvartsman to a seminar at NITMB. Stas did his undergraduate at Moscow State University before moving to Technion for a master's, and then went on to do a PhD at Princeton in applied mathematics and dynamical systems. And then he decided that he will do biology after this, which has stayed with him for a long time since. Went on to do a postdoc with Doug Laffenberger, and then started his own lab in 2000, 2000. And it's very relevant to NITMB in a sense that before he started his lab, he had never picked a pipette. Legend has it, he still hasn't picked a pipette. But his lab is predominantly experimental now, and they seamlessly mix mathematics and biology and experiments together to ask exciting questions in biology, developmental biology. He has, of course, done great work, and I think he's probably the most proud of the award that he got at Princeton for being an outstanding mentor, where his former trainees all wrote letters to nominate him, which is given once to one professor every year from all of Princeton in STEM, and he got that. And naturally, he has trained several excellent trainees who have gone on to do well, and I'm excited to welcome him to give a talk here. Welcome.
Thank you. So it's I was excited to visit. I heard great things about the place from Alex Mogilner, who came here several times. It's absolutely beautiful. So this is our building. The architecture is not what you have here, but we try to compensate by the quality of research. Okay. So I will tell you today, eventually, okay, so what's the best? Yeah, so I will tell you today eventually about some of the recent data that we collected in the project, where we look at the timing of the juvenile of adult transition in animal development. And in this project, we start all the way from molecules to networks to tissues and to the behavior of the whole organism. So I promise there will be original data. But before I get to that, I want to share with you my recently acquired knowledge about an episode in the history of genetics that I discovered completely accidentally when I found out that something that we have been very proud of was done by a 24-year-old student in Moscow in 1927. Okay, so I'll tell you about the story of this thing and how it connects to what we're doing now with addressing old questions with new tools. All right, so let's start at the beginning. The science of genetics was started by Mendel. You can singularly identify it with the publication of his paper, as you know, from high school or from textbooks. So he worked with garden pea. He selected 7 traits that bred true. It could be the shape of the pea, the color, and so if it was yellow, it was always yellow. But then he started working on hybrids. And if a yellow pea was crossed to a plant, was crossed to the plant that was giving green color, one of the traits disappeared, okay, in the first generation. And then it reappeared in the second generation, where he crossed the progeny, where he crossed the hybrids. And he coined terms dominant and recessive. These are original Mendel terms, so he did not coin the name gene. And the main contribution was statistical analysis of the fraction of different phenotypes. So from this, he understood that there are discrete entities that somehow influence the phenotypes. So this was the first experiment in genetics that, of course, followed the millennia of agriculture and animal breeding. But this was the first time that the person had done a controlled experiment, looked at the fractions, and understood what these fractions are actually doing it. So this was the person who failed to pass twice the exam to become a high school teacher. And so when we select students, so we should take their GPA with a grain of salt. So by all accounts, Mendel was an extremely modest person, but it is clear that he understood what is it that he's holding in his hands. So the paper was published in 1865. It is, I think, 6 years since the publication of, since Darwin and Wallace announced the birth of evolutionary theory. So I think that when he started these experiments, he did not know about Darwin. He definitely knew about evolutionary theory because in the introduction of his paper, he writes, so this is the only right way by which we can reach the solution of a question, the importance of which cannot be overestimated in connection with the evolution of organic forms. So here is the Augustine monk writing about evolution. Apparently there is no conflict, okay? All right, very good. So he published his paper. It was published in Proceedings of a Local Naturalist Society, and then it was forgotten until 1900 when it was rediscovered simultaneously by 3 or 4 different people. So the first thing that happened when Mendel's work was rediscovered by De Vries and Chermak was that the ratios of phenotypes and traits, so the transmission genetics, was confirmed and validated in a whole bunch of experimental systems, in plants, in animals, and then people started using in different organisms. So of course, at the time, genes, so this is the term that was coined by either De Vries or Bateson, I cannot recall now. So at the time, this was something completely abstract. Nobody knew what it was. And then in Columbia, Thomas Hunt Morgan, working with a stellar crew of 3 undergraduates, started looking at ratios of different phenotypes. And they noticed that deviation, so they confirmed these ratios largely in a number of experiments, but they also noticed very important deviations from these ratios. And this is how people understood the genes are arranged linearly on chromosomes. And in fact, there were groups that showed completely independent inheritance, and these were genes that were on completely different chromosomes. And the ones that showed different extents of deviation from expected Mendelian ratios were in the same chromosomes. And in fact, there turned out to be 4 of these groups, and the 4 groups corresponded exactly to the number of chromosomes. So this was the first time that the physical basis of heredity was established and connected to cell biology. Of course, these experiments could not have been done at the time of Mendel, and it might as well that they waited, 40 years, because at this time, cell biology made progress, chromosomal dyes were present, and one could actually connect something that you see in a cell to a phenotype that you see in the whole organism, connecting to scale. So at the time, and we're talking about, let's say, 1910, 1915, genetics was the youngest branch of biologists. At the same time, Darwinists were already established scientists with long beards. They were examining fossils. They were looking at different homological sequences, and the power of the argument very strongly depended on the eloquence of the scientists. So this was not the point that I'm trying to make is that at the time it was not an experimental science, and they were far from embracing this new science. So at the time that I am describing, Mendel was gone, Darwin was gone, but Wallace, who started evolutionary theory together with Darwin, felt that it was important to comment on the state of genetics. And this is one of his quotes. So he said that Darwin, specifically, he addressed something that was known at the time as the P versus height debate. Okay, so the P, and when you look at Mendelian traits, so these are completely discrete things. You are either one or the other, whereas in height, there are quantitative variations. So this is what Wallace was writing in 1908. He said that Darwin did not exclude discontinuous variations, but he soon became convinced that these were quite unimportant. These curious phenomena may be of interest as furnishing materials for the study of the theory of heredity, but they have absolutely nothing to do with the origin of modification of species. So this is 1908. So this argument was resolved mathematically by the young Ronald Fisher shortly after he graduated from college. So using a remarkable paper that's very, very difficult to read, but using arguments that use central limit theorem and some assumptions about mating in the population, he showed that if a trait is influenced by a handful, 3, 4, 5 different traits, that very soon you start looking at, you obtain bell curves. Okay, so at least mathematically, there was no contradiction. So he said that the phenotypes are established by a handful, so the phenotypes such as height or weight, established by several loci of small effects, and this turned out to be true. But the establishing that this is true had to wait to the genomic era. We're not there yet. Okay, so we're now in 1918. So this argument was resolved mathematically. The second argument that was the most difficult to address, that was raised by Darwinists, was that mutants that are studied by Mendelians and Mendelists and Morganists are actually laboratory artifacts that have to do with some variations that arise, some changes in the organism that arise because they're kept in the laboratory conditions, or because the animals or are kept in captivity, or because plants are raised in conditions. So this is clearly a catch-22 argument. So to study this, you need to do a controlled experiment. You can do it in the lab. Somebody can argue that you expose flies to ether, and this is why they come out deformed and the eye colors are very different. So this is argument number one. The second argument is that if people looked at natural populations, mutants were extremely rare. And the argument that a mutation has anything to do with evolution was immediately met with a counter argument that something like this would be immediately, especially if it's disadvantageous in any way, shape, or form, would be immediately killed off and not be able to contribute to the next population. So this continuous to continuous variation was resolved in a clean way, at least mathematically, but the argument about the relevance of this for the evolutionary progress was still unresolved. So I will tell you how it was resolved in a completely incontrovertible way experimentally, and where it was resolved. All right. So the work that I'm about to describe was done in the 1920s in Moscow in the Institute of Experimental Biology. The institute was run by Nikolai Koltsov. Koltsov was a zoologist. He was trained before the revolution. He visited Europe frequently, met Morgan and Boveri and Goldschmidt, so he was plugged in. At the same time, he was opposing monarchy in Russia, supported student movement. So after the revolution, he could continue his work, and there was actually an institute where he was doing this work. He was well known, and and at some point foreigners became brave enough to visit Moscow. In the early 1920s, they brought Morgan's book that was translated and used to study genetics. So because of the two revolutions actually, and because of the bloody civil war, Russian scientists were completely behind with genetics, so they had to catch up. So bringing the Morgan's book was very important. But the institute where Koltsov worked was extremely interesting. So he was invited to give a report about this unusual institute in science in 1924. And he writes the following thing. So this is 1924, okay? So in every science, the best results are obtained when the same theme is treated with two quite different methods belonging to two different scientific branches. So this was not very common in the beginning of the century. Therefore, I have put together in my institute cytological, which is cell biology, biochemistry, physiology, behavior, genetic, and eugenic sections. Okay, so if you substitute genomics for eugenics, you would have a description of a decent biology department today. So this was an example of doing interdisciplinary research. And I will tell you about one of the things that was accomplished there that still has an impact in the way that we think about natural populations and in the way that we do science today. So the genetics laboratory in the institute was headed by a person about whom I will tell you much more. So his name was Sergei Chetverikov. So he was Koltsov's second in command. He was around 40 years old at the time. So he assembled a group of 10 zoologists. So here they are. They were all in their 20s. And here they are sitting at the steps of the house at which the students of the Moscow Biology University were doing their practicums for many, many years. I studied there in the chemistry department, but many of my friends sat exactly on these steps. Okay. So they took apart Morgan's book. They translated it into Russian. They formed a very interesting way of discussing results. It was called Droz-Saor. Saor in Russian is совместное орание, or joint shouting. So it's Droz-Josh. And so in this format, any person could interrupt any other. So they had to be a collection of people who are not offended by. So by the way, feel free to interrupt and ask questions at any moment. So any person had to be able to read in 3 different foreign languages. They, depending on who knew which language, they would present either short papers or longer chapters of books. So they studied literature, and they zeroed in on one specific problem, which was the conflict of evolution, the argument that the mutations are monsters and there are no mutations and there are no variants in natural populations, and genetics that could be studied in a clean way in the style of Mendel. And they managed to do both field work and laboratory studies, and this is the experiment that I'm going to describe. Okay, so what was the experiment? So first of all, I forgot to tell you that in 1921, Hermann Müller, who was one of the 3 students of Morgan, came to Moscow and brought to Moscow about 20 or 30 lines that were established in the fly room in Columbia. Okay, so using these flies, it was possible you could do genetics, and by complementation, you can, for example, check whether the phenotype that you are studying corresponds to something that was studied before. So this was already useful. So even when this is the importance of traveling to places that are less fortunate than others and interacting with scientists in difficult circumstances. Okay, so independently of political nonsense, scientists should still talk to each other independently where they are. Okay, so here's the experiment that was done. So if the argument was that what Mendel was studying is an artifact, then obviously if you walk out of the lab and you find something in the wild, you can say, okay, this thing is actually escaped in the lab, so it's probably there. So these guys, so there were 2 people or 3 people that traveled about 1,000 miles south of Moscow to Gelendzhik. So this is the place, this is not quite Crimea, but this is very close to Crimea. And they collected 300 fertilized Drosophila melanogaster lines that were 300 Drosophila melanogaster females that were already fertilized. They brought them by train to Moscow. So I took these trains in the 80s and early 90s. It is, I would not recommend it, okay? So thinking about how they were bringing these flies from there to Moscow it's very interesting. So I'm trying to imagine this journey. Okay, so basically they brought them to the lab and they did an experiment that was identical to Mendel's experiment. Because these were individual lines, they generated the hybrids. And then, so they did not generate hybrids. They got the progeny, and then they crossed the progeny to itself. So they did the brother-sister matings. And in this experiment, they took just a couple of weeks. So you can completely rule out emergence of mutations or emergence of variations because of this manipulation. Even though that country is a highly mutagenic environment, okay? So they discovered that each line harbors 5 or 6 distinct mutations, okay? Mutations that could be recognized by morphological changes, okay? Some of these changes were subtle, like, for example, the appearance of extra vein on the wing or the appearance of extra bristle on the thorax. And some of these changes were absolutely enormous. So they discovered that in F2, they observed, for example, antenna to leg transformation. And they had flies where there was a haltere to wing transformation. So these are homeotic transformations that apparently take the fruit fly and start converting it into a cockroach. Okay, this is obviously not what is happening, but this is an illustration that these populations are saturated by mutations. So this was the experiment that was started in 1925. It was published by these students. So they published single author papers. And in 1926, Chetverikov, who was the PI of this group of young people, wrote a paper of which we are going to have a centenary this year. The paper was titled On Certain Aspects of the Evolutionary Process from the Standpoint of Modern Genetics. So his main point, so the opening sentences of the paper, they speak directly to the very hostile reception that Mendelian genetics received from Darwinists. And today it's completely unimaginable, this conflict. It's like a conflict between peas and carrots. Because today, the most convincing arguments for anything that has to do with evolution has to do with the analysis of the genomes, right? So, of course, it's supported by fossils and by developmental studies, but this is one of the strongest evidence that for relatedness of things. So this is the main conclusion of the paper. He said that natural populations are highly heterogeneous with numerous spontaneously arising mutations. So he used a different term. He used the term genovariation, maintained mostly in a heterozygous state, hidden from the eye in phenotypically identical but genetically diverse individuals. So the answer to the argument that we never see mutants in the wild is that because they're hidden so if these are recessive mutations, they're hidden. Of course, we need to understand that they phenotyped what they could phenotype at the time. So it was a morphological phenotype. So there are many more variants that are there. So any mutation that one could potentially study was present in, as they called it, genetic milieu, and something that we call today a genetic background. So this explains that even in populations that are completely homogeneous phenotypically, there is an immense genetic heterogeneity. So these were extremely important ideas. But one way or the other, this was the very first experiment in genetics of natural population. Okay, this is how it started. So if you know about the experiments of Dobzhansky, so they came out of these ideas. Dobzhansky initially, before he moved to the States, worked in Kiev, in Ukraine, but he visited Koltsov Institute and he participated in the discussions that led to this first experiment in genetics of natural populations. So everything was going great, but you need to remember where everything was happening. So in 1929, Chetverikov was arrested and exiled from Moscow. He was prohibited essentially permanently from returning to Moscow and returning to any of the 11 large cities of the Soviet Union. So he could not travel to Ukraine. He could not travel to republics. He had to make a living. So first he was sent to Sverdlovsk, which is the place where the Tsar's family was executed in 1918. And initially he worked as a consultant in a zoo. Okay, so here is the person who is really an amazing scientist forced to make a living as a consultant in a zoo. Then he got a slightly better job. He worked as a teacher in a vocational school that trained people to deal with pest control. So he knew a lot about insects. Eventually, he was allowed to return to science. So he worked on spider silk because of connections, and silkworm because of connections to military, because you make parachutes with these stropes. And in 1948, he was when Lysenko triumphed over not only genetics but Darwinism in the Soviet Union, he was permanently banished from science. Okay. Several of his group, so the group was disbanded. One of the person eventually ended in jail and was executed. The other one got in jail and got out. So the whole thing was dispersed. The only good thing, so this amazing trajectory was cut short. But one of the group members, his name was Nikolai Timofeev-Rysovsky, left Russia. So he left under very interesting circumstances. In 1924, Lenin died, and the Soviets invited Oskar Vogt, who was a neurosurgeon, to come and dissect the brain of the founder of the greatest state in the world history, to see what is it that was special in the brain of this mass murderer. So Vogt came to Moscow. He established the institute that studies until now. It's called the Preparation Number 1, which is Lenin's brain. And he was very interested in genetics of psychiatric disorders. He did not know genetics, but he knew that the people that he is interacting with know about genetics. And he asked who can be sent to Berlin to start a genetics laboratory in his institute. So Timofeev-Rysovsky and his wife, who was also a member of this group and also a scientist, left there, and they were in their mid-20s, so the age or younger than the age of a graduate student. So he became a leading geneticist working on 3 problems. So the first problem was the genotype-to-phenotype connection. The second problem was about the origin of mutations. What kind of factors control how quickly some mutations are happening? And the third one was on the genetic structure of natural populations. So he worked in Berlin So he has an interesting life history in and of itself. So it's a topic for a separate discussion. The point, what is important to us is that working in Berlin and organizing the discussions of the style of Chetverikov's group, he met young Max Delbruck in Berlin, who was at the time looking for new problems, for new physics problems in the context of biology. So Delbruck felt that he missed the birth of quantum mechanics, and he was looking for something new, and encouraged by Bohr, he was looking for new for new physical principles in living systems. So Timofey Rysovsky, who was a geneticist, and Delbruck, who was who was a theoretical physicist, they worked together with Zimmer, who was a member of Timofeyev's group in Berlin. Zimmer was an experimental physicist. He was a radiologist. He worked on the effects of radiation on the frequency of mutation, and he was one of the first people who understood that you actually need to measure the dose at the time, because before that, the intensity of radiation was measured by an experimentalist placing a hand in the bin and recording when it got red. So this is not, okay. So they wrote a paper that is called either a three-man paper or a green pamphlet that was published in the Proceedings of the Scientific Society of Göttingen. And this is the paper on the nature of the mutation, on the nature of the gene structure. So the paper was published in 1935. Delbruck later on was joking that by publishing it at this place, they gave the paper the first-rate funeral. But this pamphlet, so it was published in a very small number of copies, was sent to a number of people in Europe. And at some point, Ewald, who was a theoretical physicist, gave it to Schrödinger, who at the time was already in Dublin. So Schrödinger was thinking about these problems. He was extremely impressed by this three-man paper. So in this paper, this is one of the first places where it says that a gene might, which is something nebulous and abstract in genetics, must combine stability and ability to change. Most likely it's a molecule. The terms polymer appear already in this paper. Schrödinger converted it into aperiodic crystal. So this book, so he gave a series of lectures, largely based. So the book is written in extremely poetic style. It says that living systems are very different from non-living systems, and we can predict the life of a sparrow with much greater accuracy than the lifetime of a radioactive atom. So this book brought many physicists to biology, and it popularized the ideas about the structure of the gene that can be traced all the way back to Koltsov to the West. Europe at the time was what Europe at the time was. Delbruck moved to the US on the Rockefeller Fellowship. And initially, he went to Caltech to work with Morgan on flies. So he very quickly understood that flies are way too complex if you want to understand something about the structure of the gene. And he started interacting with Salvador Luria, an immigrant scientist from Italy, who read the green pamphlet. And they started interacting already during the war. And in 1943, they published a trailblazing paper that addressed the following question. So a population of bacteria was exposed to bacteriophages. After they were exposed, some bacteria died, some bacteria did not die. And the question was, so you could show what is the origin of this resistance to bacteria. Is it something that pre-exists in a population of bacteria, or this is something that is induced by the virus? So notice the style of the question is very similar to the style of the question that Chetverikov was asking, whether the populations are already filled with these conditions. So you know about the fluctuation test, and they show that by looking at the fluctuations of the number of the sizes of bacteria, they could at least statistically show that mutations exist before, and if mutation arises before, then it can protect you from the phage. It's not the phage that is inducing this resistance. Okay, so this paper was the start of the phage group that eventually led to the Nobel Prize. So initially these were very young people that eventually in 1969 resulted in a Nobel Prize for Delbruck, Hershey, and Salvador Luria. What is interesting is that the very first graduate student of Salvador Luria was Jim Watson. Okay, The rest is history, which is not ideal, but it's history and brings us to the present day when we talk about genes in terms of DNA and in terms of, okay, so I'll skip 50 years or 70 years. Okay, so where are we now? So if you think about the experiment that was done by Chetverikov and his students, they went into natural populations and surveyed phenotypes, and they tried to infer heterogeneity of genetic composition without knowing what the gene actually is, right? So today there is a resource that's called Drosophila Evolution Over Space and Time, where people take they sequence, hundred or thousand of different Drosophila in different populations in different parts of the world, and you could study adaptations. Of course, this is not limited to Drosophila. There are genomic resources like Biobank that contain 500,000 healthy individuals in the UK. There is All of Us, which is an American version, the Finnish version. There are genomes of the population that have autism spectrum disorder. So this is an example of surveying the genomic composition of the population that can be traced back to these early experiments. So so when it comes to analyzing genetic makeup and sequencing, we are getting better and better and better and better and better. Okay. One of the main challenges in and because in some cases we know how these genes work, there is a clear temptation to predict what the phenotypic consequences based on what we know about the molecules. However, because it turns out that if you look at the disease alleles that were identified by sequencing affected individuals, many of the asymptomatic people in UK Biobank and All of Us are carriers of these alleles. So obviously, the penetrance is not 100%.
There's a question online.
Yep.
Why was Drosophila too complex as opposed to phages for the experiment?
Because of the turnaround time, because of the size of the genome. Okay, very good. So one of the key issues has to do with penetrance and expressivity. Penetrance is the chance that a carrier will actually display the phenotype. Expressivity is how severe this phenotype was. So whether or not you will have a heart defect and how severe it will be for heart function. And this is a title of the paper that was given last year by Bruce Gelb, who at the time was the president of American Society for Human Genetics. So these are some of the key questions that the community of human geneticists that is completely overwhelmed by sequencing data that is coming more and more is struggling with. Okay, so finally, I will give you one example from our work so that you don't get an impression that I'm pretend to be a historian of science, which I'm not. Okay, so I learned of all of this because at some point I discovered the papers of this group of Chetverikov and Koltsov, and I realized that some of the ideas of penetrance, of genetic background, stochastic fluctuations existed already 100 years ago. So one of the themes in the lab, and this theme was actually started by Yogesh and his academic brother when he was a graduate student in my lab, has to do with mechanistic understanding of developmental defects. So right now we're addressing this problem, Mendelian developmental defects. Okay, so we're addressing this problem in two different ways. So first of all, in a line of research that I will not describe today, we have established the simplest imaginable model for a structural defect. So this is a model where defect can be reduced to a single cell. So the cell is either there or not there. So it's a discrete phenotype, completely in the style of Mendel. And it's a partially penetrant phenotype. We can watch how this phenotype is emerging as a function of time. Okay, so we can study dynamically the connection between the genotype and the phenotype. So one of the main challenges so the reason why issues that have to do with penetrance and expressivity are so difficult to study, because genetics across organisms, by and large, still works with finite phenotypes, with endpoint phenotypes. So we need to, I think that by learning more about the ways in which the phenotypes actually arise or do not arise, we will learn more about we will get smarter in connecting genotypes to phenotypes. And then the second problem that I will highlight today has to do with changes in timing, because you can either do something incorrectly. Let's say you solve a problem on the test incorrectly and you are marked down, or you can solve this problem in 10 times the time that is allowed on the test. So such a thing can display as a variation in the time in which you accomplish the task. So in the language of dynamical systems, you end in the same attractor, but you end there on a completely different time scale, either very fast or very slow. So one of the phenotypes that I got interested in individuals with conditions that we started studying already when Yogesh was in the lab was that they're characterized by what's called disorders of puberty. And the puberty can have late onset, and the puberty can have early onset. And in fact, so this is a very impressive graph that shows us the age of menarche in women from the end of the 19th century, where it was approximately 17 years, to where we are now, where it's 11, 12 years. So there is the overall population-wide trend that has to do with the decrease of this timing. So it largely has to do with obesity. The point that I want to make is that this is a phenotype which is quantitative. So it's not like a P, it's like height. But instead of the height, it is the time when something is happening. So I will tell you how we study this. Very good. Okay. So what is happening when we hit puberty? Okay. So puberty is a transition between the juvenile and adult form of the organism. It's a transition between periods of life when we are growing and not reproducing to the time when we're capable of producing or not growing, if you don't count increasing waistlines. Okay. So what is happening? So there are changes of two different types. So this is the change in the growth velocity. So this is the spike at puberty, and then eventually the growth velocity goes down to zero. And puberty is associated with changes in the dynamics of the hormonal circuits that control the circulation levels of sex hormones like estrogen and testosterone. So these things have to oscillate. They oscillate on the time scale of an hour right now. So there are cycles in the production of gonadotropin-releasing hormone, which ultimately talks to our gonads and controls the production of sex hormones. So at birth, there are oscillations. Then during childhood, these oscillations disappear. And then somehow we enter the regime where there are oscillations, where there is pulsatile production of these hormones, and we stay there. So it seems like we're walking into a new dynamical regime in the production of hormones. Okay, so this is the, okay. So this is some of the circuitry that is involved. So what is interesting about it is the same thing that's complicated about it. So it's a change that involves the entire organism. You cannot study it by studying a single tissue, a single cell. You need to resolve all of these things, but you need to think about the entire system. So what is happening is that at some point as you grow, you increase in size and you accumulate a sufficient amount of fat tissue. Fat tissue secretes hormones, one of which is leptin, which many people know as a satiety hormone. Leptin talks to the brain. The brain ultimately talks to a part of the brain that's called the hypothalamus. And it is in the hypothalamus that there are oscillations in the production of gonadotropin-releasing hormone. The hypothalamus talks to our pituitary gland, and our pituitary gland talks to the gonads. Okay, and so you need, and two things happen simultaneously. You tell the organism to stop growing, and this corresponds to changes in the growth zones in the bones. This is how the height is controlled. And then we start producing mature gametes, eggs for a female, sperm for male. Now, all right. So, and of course, delay in puberty is problematic because just don't think about a human. If you are an organism, eventually you want to become adult because you need to reproduce at a certain point. So if you are, for example, extremely lean, anorexic, you might never hit periods. Okay. On the other hand, if you hit puberty prematurely, this can be just as problematic. So you can be stunted in your growth. Okay, and there are all kinds of psychological problems, and in fact, it shows that epidemiological studies that yes.
Is that true? If I take an organism and I keep it much too thin, can I actually experimentally delay?
Of course, of course. So, for example, during the siege of Leningrad, many people got stunted, and there is a very famous example of in human populations with hunger in Netherlands. So during the war, so there was northern part of Holland was cut off by the so the Allies were coming in, the Germans cut off the supply of food, people just were stunted, and interestingly, it persisted for 2 generations.
I understand that.
So the answer is yes, you can.
I guess what I'm interested in is the bounds of the system. If I accelerate,
this is precisely, I'll tell you how we study. Okay, so these are the kinds of questions that we're interested in exploring. The challenge is that you need to study it at the level of the organism. Human and the mouse is a very simple organism. Okay. I cannot see the time. I have 15 more minutes.
Yes.
Okay, this is fine. Okay, so it turns out. So this is sort of imagine that this is a Vitruvian man and this is a Vitruvian fly. So it turns out that even though an insect is completely different from a mammal, there is a lot of similarity in the neuroanatomical circuits that control the hormonal regulation in the animal. So we have liver and fat tissue. Flies have something that is called the fat body. Okay, we have blood. Flies have hemolymph. We have a pituitary gland. Flies have something that is called the ring gland that serves exactly the same purpose. So anatomically, you can study the same. So and then there are parallels between different cell types that you can convince yourself of by looking at single cell profiling. All right. So you can. So the circuits are controlled. If you look, okay. The molecules are also controlled. Okay. So this is the schematic of the development of the monarch butterfly that goes from an egg to a feeding larva that hatches and grows and grows and grows and grows. Eventually, it becomes a chrysalis, and then it becomes a butterfly. So it turns out that if you look at the butterfly, and if you look at any insect for that matter, the size of the adult is completely set by the time when it decides to form a pupa and chrysalis. So here is that it is in a growing and feeding state. Here it is a non-growing and feeding, but the feeding is just for maintenance, okay, and reproducing state. So this is absolutely a critical transition. So the decision to pupate, to decide to stop eating and start remodeling itself is juvenile to adult transition. Okay, what is this thing controlled? Okay, turns out that at the point when it is happening, there is a surge in the circulation level of a molecule that is called 20-hydroxyigdicin. Okay, so if you don't like organic chemistry, too bad, but this is the molecule. So it's a hydrophobic molecule that can pass through the cell membranes. And this molecule is acting as a ligand, intracellular ligand, for proteins that act as transcription factors. Okay, so if you know about even the simplest genetic systems like the lac operon, so there you have an organic molecule like allolactose binding lac repressor and controlling gene expression. So here you have 20-hydroxyigdicin, also an organic molecule, acting as a ligand for a protein which changes the way in which this thing interacts with the DNA. Okay, so this thing controls the expression of genes that are responsible for remodeling. What is absolutely remarkable, and I had no idea about it just until a couple of years ago, is that both igdicin, so igdicin is also derived from dietary cholesterol, just like our testosterone and estrogen. And these molecules are just a couple of organic reactions away from each other, to the point that you can be giving human estrogen to insects, to butterflies, or to Drosophila, and you will accelerate these transitions. It is not by chance that these effects are observed. It happens because they bind to exactly the same transcription factors that bind to highly conserved sites in the enhancers of the genes that control remodeling. So the anatomy is conserved. The functional nature of the decision is conserved. And the molecules that implement this decision are conserved. So this is a very interesting example. Okay. Okay, how is this studied? And this is something I must say that when I started reading it, it's not a very, so it's a literature on insect physiology. It's not very difficult. It's not very easy to get into this. And I started interacting with Alex Shingleton, who is a faculty in the University of Chicago here. So he told me a lot of the things that I know. So how is this thing studied? So it is, so remember I told you that in humans, you reach a certain mass. And at that mass, something is happening. So what you can be doing, you can be looking at the mass as a function of time, okay? You can also be looking at the concentration of hormone in the blood. So the critical people of you can ask me, where are the error bars in these measurements? They're not here because there are no error bars. So these are extremely, so in principle, you can say it's easy to measure the mass, it's easy to measure the concentration of this organic molecule in the hemolymph. These are disruptive measurements. They are obtained by stopping the experiment, grinding a whole bunch of larvae. So it's, in theory, it's easy. In practice, it's not something that you would want to do on your own. Alternatively, so you can study the mass, something that controls the hormone. You can study the hormone. Alternatively, you can study individual events. Okay, so this is a petri dish with a wandering larva that you see here. It moves around. It eats. And at some point, it will, the video will be sped up. It will realize that it has eaten enough, and it will convert into a larva. So you can detect these events. And this is something that can be measured very precisely. And this is something that can be measured at the level of the individual. So how is this actually working? So insects, so we are not studying, so butterflies are beautiful, but we study it in Drosophila because of the genetic power of the system. Flies have a fat body that secretes a hormone that talks to the brain. So this hormone is insulin. And then it talks to a ring gland, which is an endocrine gland, which is an equivalent of our pituitary. So there are just 4 neurons that, actually 2 neurons that innervate the pituitary gland. This endocrine gland secretes ecdyson that upregulates the production of ecdyson in itself, which generates an irreversible switch in the circulation levels of the hormone. And this is how the animal enters, gets to the point of no return. So this is the circuit. So the circuit is quite complex. So every one of these arrows contains, dozens of reactions. So I can wave my hands and say that the system is quite understandable. Mechanistic modeling is out of questions at this point. So realizing that mechanistic modeling is out of questions, we decided to approach in an engineering way, where we would perturb the system, look at the responses to time-dependent perturbations, and identify the time scales and the thresholds in small numbers and use it as a predictive model of what is happening. Okay, so how did we do it? We do it because we know that one of the molecules, so this is the brain, this is in gray here, this is the pituitary gland. So one of the molecules that is involved in production of ecdyson, of this sex hormone or maturation hormone in the gland, is an enzyme that we have been studying for a very, very long time. In fact, from the time when Yogesh was a fledgling scientist in our lab. So this is a molecule that's called MEK. We understand a lot about this molecule. We know a lot about the mutations, about human conditions in cancer and developmental disorders. And this knowledge, together with advances made by other people and students in the lab and other laboratories, allow us to place this enzyme, which here is shown in cyan, under control of light. So we can perturb this enzyme by light. So the point is that this enzyme is introduced into the genome of a fly in a constitutively active form, but the active site is flanked, but the enzyme part is flanked by the 2 separated and photodimerizable and photodissociable forms of the protein that are called DRONPA, okay? So in the dark, these domains dimerize. In the green light, this enzyme is active and gets access to its one and only substrate, which is ERK, which is something that controls many things inside the cell. Okay. So this is something that we started doing when Alina Patel was a graduate student in the lab, who was a student recruited by Yogesh. So you see the continuity here. And then we learned a lot about how this enzyme changes across evolution, and we can control it quite precisely now. All right. So here is the first experiment, and believe me, I'll take you through the rest of the day. So in the dark, we can detect events. We can have 100 of these larvae that grow and eat. For each larva, we can detect the time when it becomes a pupa, which is a point when it stops moving. And this happens approximately 4.5 days after the egg was laid. In light, we offset it by a day. Okay? You offset it by a day. What does this mean? It means that the animal decided to stop eating 1 day earlier. If it stopped eating, it should be shorter. It is shorter by approximately 10%. So this stuff just came out in PNAS a day or 2 days ago. There are error bars there. I don't want to bore you with the details. So the pupa are shorter and the animals are shorter. So we can control when it does something. Very good. So now that we have a system that is sensitive to external perturbations, we can try to evaluate its mathematical parameters. So what we know just qualitatively is that mass is the signal that is sensed by the brain. Brain talks to the gland that eventually can turn something that autoregulates. So we wrote the simplest possible dynamical system where we have the mass that says that has 2 variables, mass and hormone. Mass is increasing at a constant rate until your hormone concentration crosses the threshold. Past this point, it is not changing. And concentration of the hormone is positively regulated by the mass. There is a clearance by the tissues that uptake the hormone. And then there is also autoregulation, because we know that there is autoregulation from genetic studies. Very good. So, so let's see what happens at constant mass. So when mass is when mass is 0, this equation has 2 roots, one 0 and one that is positive. Past a certain threshold of mass, this parabola no longer crosses the horizontal axis, which means that there are no steady states in the system. And this corresponds to an event that is called the finite time blowup, which is something that can explain very sharp increase in the concentration of the hormone. We're not saying that there is no. So eventually, of course, there are no infinities in real life. Okay, eventually, new terms in the equation kick in and balance it. But this is something that can potentially describe the early part of the transient. And we think that this is something that explains the surge in the hormone levels. So far, so good qualitatively. So qualitatively, you grow, and there is a surge when you cross a certain threshold that corresponds to a bifurcation in this model. If you starve the animal and you never reach the threshold, you are stunted and the surge does not happen. If you stimulate it earlier, surge will happen earlier and you will be shorter. Very good. So what are the actual parameters of the model? So if we rescale, if we make the model dimensionless, we rescale the mass and the hormone by the values at the bifurcation point, we realize that there are only 2 dimensionless groups. One of them is the ratio of time scales, and it's the time scale of the clearance of the hormone from the circulation and the time scale to reach the bifurcation point when you grow at a constant rate. And the second time scale is the ratio of the 2 thresholds, the threshold at which you decide to stop eating and the bifurcation. Okay, so you overstep. So it's like when you build a nuclear bomb, you need a mass that's greater than the critical mass. The question is by how much do you increase? So by how much heavier is your bomb than the critical mass? So there are only 2 parameters. We can also add the effect of stimulation. And this introduces a 3rd parameter that tells you how sensitive is the system to our perturbations. We know that this is sensitive, we just don't know how much. Then we can design different stimulation protocols, different pulses. So we can stimulate constantly, we can stimulate early, late, place these pulses wherever we want, and we can measure the advances in hours of transition to metamorphosis. And we can uniquely identify parameters of this model. We don't have a proof that this is unique, but the model is simple. We have scanned the 3-dimensional parameter space, and we have these parameters with quite tight error bars. Okay, so let's look at these parameters. So the first thing that we need to notice is that epsilon is approximately 1, which means that there is no separation of timescales in the system. Initially, I had hopes to make the model even simpler so that we could handle it using singular perturbations. Turned out not to be the case. The threshold is approximately 1.5. So you overstep your critical mass or your critical hormone concentration by approximately 50%. And sensitivity to the external stimulus is 1, which is good. So the fact that these things are all approximately 1 means that all rescaling was done correctly and everything is the way it should be. So now we have this model. We can predict the responses of a system to the stimuli that were not used to estimate parameters. So we identify the regime in the model where the advanced transition to metamorphosis displayed non-monotonic behavior at the time where the pulse is placed. So early on, it's very easy to understand why this is happening, because initially the animal is too small to sense that you're trying to hasten it, right? It's like teaching calculus to a 3-year-old. So unless it's Gauss, it will fall on deaf ears, okay? And then eventually, at late times, you will also not help it because it's already doing things on its own. So the sensitivity has a peak in the middle. So the dashed lines are predictions. The solid lines are what is happening. The agreement, there is non-monotonic behavior. Question, it's even quantitatively okay. Very good. So finally, in the last couple of minutes, I will tell you that what we are doing right now in the lab. Because in transition to puberty, you not only start producing gametes and you not only stop growing, but there are all kinds of changes. There are behavioral changes. It's a very rich phenotype. So we built a device. So people in the lab, so Dennis Farberg, who is actually was a technician in the laboratory of Ilya Rovinsky in Chicago, brought his expertise in working with C. elegans to the lab. And with his help, so he is a graduate student in the lab, and with another graduate student, Changyang Wang, we built a device that is called the oxodrome. Oxo is growth, drome is when you run together, like syndrome, hippodrome. So, so this is a device in which 96 animals can be studied, can be imaged in their development from the time that the egg was deposited to the time they go through their life changes. It is quite cool. So the egg, here it is imaged. Nothing is happening, the time is going on, and then computer picks in that there is motion. It's like a detection sensor. So we can very accurately measure when it hatches from the egg. This is pupation, so it moves. Moves. At some point, it will stop moving, and just like you detected that the motion was detected, you will detect that the movement disappeared. Okay, so this has become a pupa. Eventually, in a couple of days, in the same well, there is an eclosion, nothing is happening, and then all of a sudden, you will see an insect that came from this thing, okay? So we can measure, and these are the, so this is now what we're using for phenotypes. So these are the tracks of the newly hatched larvae of a wild type, and this is a mutant, so this is NF1, so these are the flies that have a mutation in neurofibromin type 1, which is a founder, which is one of the most common diseases in the rhasopathies, that class of diseases that we started studying with Yogesh. And you know, when somebody says that these are the baby's first steps, so these are the larval first crawls, okay? So this is essentially it. This, I told you about the story of a conflict that was resolved with the help of mathematicians and with the help of zoologists in the first experiment in the genetics of natural population. I think that the genotype to phenotype map is something that is interesting to many people in the room. Most of the studies in the literature look at the genotype, which can be studied and understood very precisely with sequencing, and phenotype as an endpoint. Looking at development, I think we will gain additional insights. And I told you how we're studying in the context of transition to metamorphosis. So these are the people who did the work. So this is Rob Marmion, who is a postdoc in the lab. This is Changyang Wang. So she's a third-year graduate student in the physics department. And this is Dennis Farberg. And this is a Kandinsky-style rendering of, 20 different larvae as they move. So thank you very much. I'll be happy to answer any questions. Yes.
Super inspiring. So, and the optogenetics, so cool. I have two questions, both from experiment point of view. So first, when I was learning fly genetics, I was told the light, dark control is very important for the fly development, right? 12-hour light, 12-hour darkness. So when you do the optogenetics, you keep them completely dark or completely in blue light, it's going to affect the development.
Okay, so this is why this thing is not activated by blue light.
So this is not,
It's activated by green light.
but I mean, it's also you keep in complete dark.
Yeah, but
right?
we know that.
Circadian genes are not going to be involved.
Yeah, absolutely. You're absolutely right. So there is another way to stimulate the same pathway that is based on a different optogenetic system that Yogesh helped develop, and he was a graduate student that's based on optosource. That thing is not as good.
So you're saying the blue light is
So
going
the fact that there is no blue light.
Because I noticed when I shine too much blue light to the fly, they do not like it.
This is why you need a different optogenetic system. Yeah.
And the second question is about yours looking them individually, right?
Yeah.
Have you ever think about interlarval
So this is this is extremely important. So when you people who work with insects, right? You know that they collectively churn food. And when you churn food, you help each other. And in fact, larvae can dig tunnels. Yeah. This is just studying the system under the conditions where we can study it. We think it's already sensitive enough. But the fact that there are collective effects is unquestionable. We're very interested in it, but there is just not enough hours in the day. One can certainly, but I think with the advances in computer vision, there will be absolutely no problem to study pairs of larvae, triplets of larvae. You just need to successfully detect who is who.
Yeah, they do cooperative digging, right?
So yeah, absolutely.
They have social behavior.
Yeah.
They help each other.
Yeah, of course. But there is social behavior arises as a result of interactions of what is there in the individual. We have, what we have is not perfect, but I think it is already rich enough to be informative. You are absolutely right. So it can easily happen that a phenotype that we will discover in individual larvae will completely disappear through interactions, or will be amplified. I mean, who knows? But we're doing experiments where we measure the same thing in bottles, and yes.
In your very elegant talk, and my question is about the little toy equation for hormone level and mass. You have the H-squared term, which you put is, of course, the simplest nonlinear term you can put, but it seems to work very well. Yeah. Do you have a mechanistic understanding of why it is H-squared? For instance, maybe some transcription factors bind as a dimer. And
so H-squared is only to give us the finite time blow-up. The same thing would be happening with H-cube or anything like this. We know that there is an origin of that there is autoregulation, because if you express in the prothoracic gland something that gets rid of your ability to sense hormone, so then there is a massive delay. But you can, so the only thing that our combined models and experiments tell us is that this model is rich enough to account for data and make sensible predictions. Mechanistic interpretation of nonlinearity in something that has to do with transcriptional regulation is not understood even by people who study bicoid.
Right.
So in this case, where you study like transcriptional responses, so here we're studying something that is miles away in the chain of events. I can pretend that we understand, but it will be all.
And from the data, you cannot tell what exactly is the shape of because you have the experimental data on how it blows up.
Yeah, but we have data of the time of an event. So what we are doing in the lab. So from the lab of David McKay at NC Chapel Hill, we got a couple of lines that express fluorescent reporters of genes directly controlled by Dyson. So by measuring times of these things lighting up, we might be able to get closer to the questions that you are referring to. Yes. Thank you.
Did you keep the light on the entire time?
No, it depends. It depends which stage. So in the Okay, so okay, so the one where flies were 10% shorter is continuous illumination, and then there were what is it, 5 things of different pulses. Okay, we also know that if we shine light during the first and second instar, there is absolutely no effect. So there is something that developmental biologists would call the phenocritical window when you're sensitive. So that's the whole point, that you have the ability to play with this, to perturb the system at different times. Conceptually, there is nothing different. So imagine that you have the simplest imaginable mass spring dashpot system, and you don't know the mass, you don't know the friction coefficient, and you don't know the spring constant, right? So, but if you jiggle it and you watch either the frequency response or the transients, from these things you can infer these parameters. It's nonlinear, but the idea is exactly the same.
I think like if we follow the circadian rhythm and like keep a 12-hour on-off period.
So circadian rhythm definitely influences P variation, because because when you hatch, obviously matters, and so this is something that I don't know all that well, but this goes back to the work of James Truman and Lynn Ridiford, who found out with not only in flies but in Manduca sexta, that if you miss the chance to do it in this day, then you do it only a day later. So it's like you get on a train, so that leaves once a day, and if you don't get it, you get on it later. No, photoperiod is extremely important, and this gets back to Wen's question about what we keep in the dark.
Another thing I was wondering about, like, of course, the wavelength was always 500 nanometer, but did you change the intensity of light, like the amplitude of?
No, so this was done at a single amplification. Now that this is working. So first of all, I need to tell you that these experiments were done not in the oxydrome. Now we will do this in the oxydrome. So, and there we can just go to town and play with intensities and with different frequencies and oscillate and things like this. Yeah. Yes.
I have a question about the experiment where they, like with the 300 females that they transported on the train and then crossed the progeny of those females together. So you mentioned they saw some homeotic transformations?
Yes, at least 2.
Okay.
So there is one that was called tetraptera, where there were extra wings. And this is how I discovered this group of people in general. So when we were studying, so it's like the classical bithorax experiment, right, in Lewis. But when we were studying the structural defect, we zoomed in on a phenotype of the formation of extra tracheal cells. And we made a big deal in a paper that we published about the stochasticity of the phenotype and the fact that the phenotypes on the left and right-hand side of the body were completely different. Then I discovered that one of the students from this group of 10 people, Boris Astaurov, published this study with tetraptera mutation. So this was the first thing that they found. And then the second thing so this was one homeotic mutation that they discovered. The second homeotic mutation was something that is called aristopedia, which is the transformation of the antenna into a leg. It's remarkable. Yeah. So aristopedia was already known to, I think, bridges. But because they had the lines that were brought by Miller, they could show that it's allelic, what they have. This is super interesting. Yes.
I mean, given that ERK control many things, ERK levels control many things. Is it like, have you checked the dose dependent also, like how much ERK can you increase with the PSMIC, eventually that? Okay. And how early you can shift the.
Okay, how early we know, because we know that I already told you that if you do it during the first larval stages, there is no effect. During the third larva, we have a complete quantitative description of what is happening. We do not know, so we work a lot on direct targets of ERK in other systems. This is not one of them. Partly because the tissue in which it is happening is is quite small, and you cannot study it biochemically with the same, okay, yeah, with the same ease in which you can study it in other systems. So everything is happening in an extremely small place. I actually can show you how it looks. So this is the tissue. Okay, so this is, okay, so this is the prothoracic gland. This is how ERK looks in the prothoracic gland when it lights up completely. And we know that when we turn it on, it is at a higher level, but we are nowhere near quantifying it. There are other systems where we can do it much better, like the early, like the cleavage stage embryo, but this is not one of them. Yeah. Okay,